All right, welcome back, everybody. In this module, what we're going to be doing is tackling the topic of sensation and perception. Now, there's lots of different areas that we can cover when discussing this two-step process, but for the sake of simplicity and also to kind of stick in line with what many researchers have done over the years, we're going to focus our attention on one particular aspect of this process when we focus in on vision. And what we're going to be doing in this lecture is focusing on the first half of the visual process when we look at this step or this process called sensation. Now, obviously, what we've been doing so far is kind of building up to how our body is able to interact with the world through all the different neural networks and structures that we've covered. But obviously, for that process to work, for us to be able to interact with the world, we first need to know what's in the world around us. And this is where the processes of sensation and perception start to come into play. If we're looking for how to differentiate these things, sensation is all about how we're able to convert all the different energies that are in our environment into some type of neural message that our brain can then decode. That decoding process or interpretation process of those messages getting to the brain is what perception's all about. But to understand what the brain can decode, how it's able to decode it, we first want to understand how that message is getting to the brain in the first place. In other words, we want to focus our attention today on the process of sensation. As I mentioned before, we are going to be focusing our attention in this lecture on vision, but I do want us to recognize that all of our senses that we're experiencing also have some process of sensation involved in it. When we talk about hearing, we can, if we get into a sensation and perception class, look at how these tiny follicles inside of our ears, inside of a structure called the cochlea, actually bend and move in different places and at different frequencies for us to not only hear things, but understand all the nuances to sound that are out there. Things like pitch, volume, timbre, these elements to sound are all things that our ears are able to pick up on due to just the small movement, small changes in the structure of these tiny hair follicles. And if you ever do get into a sensation and perception class, you'll probably spend a couple weeks talking about how these processes work and how that message eventually gets its way to the brain so the brain can start to decode that information. This particular process is actually very complex, but not nearly as complex as our sense of touch. When we look at touch, we quickly recognize that on the skin and underneath the skin and these different areas, we have all of these different nerves, these endings that can pick up on things like temperature, changes in pressure, vibrations. We also talk about different types of touch, like interoceptive touch, exteroceptive touch. There's a lot that actually goes into that process. And when looking at all these different forms of touch, there's different cells that send messages down different paths to different parts of our brain so we can perceive all of these things. Get in again to a sensation and perception class, you'll probably spend several weeks looking at all the different components to touch, in addition to also exploring the pathways from these parts of our body up to the brain and what happens when those pathways are disrupted. Another really interesting area with respect to sensation and perception is our process of smell and our process of taste, two areas which are very intertwined with each other, even though there are some misnomers about how intertwined they actually are. Our nose does have specific receptors that are looking for information, and that information is actually sent to a very specific area of our brain called the olfactory bulb. And looking at how those sensors work, how we're able to decode the multitude of different smells that are in the environment and pick up on subtle differences in the presence of molecules that are attaching to these things is something that's still really interesting to psychologists and neuroscientists today. We also have receptors hidden on our tongue in these places called taste buds that are sensitive to things like sour or sweet, a flavor called umami, and all of these other things that we maybe take for granted, but are actually very difficult to decode. And again, in a sensation and perception class, you'd spend, in addition to looking at hearing and touch and vision, you'd spend several weeks looking at these two processes of smell and taste, just to kind of understand how these things work and how we're able to create this neural message that our brain can then decode in different areas so we can experience all these different components to our world. As I mentioned, we are going to be focusing our attention on vision. And when we talk about vision, one of the most complex things to do first is to break down what it is our eyes are actually processing. We can understand hearing fairly well when we talk about changes in air pressure that are causing our ears to vibrate. The presence of molecules are pretty easy to understand when looking at smell and taste, but unfortunately, when we talk about vision, people often have lots of misperceptions about what it is we're actually seeing. So if we want to understand vision, we have to understand the elements to the light that actually is hitting our eyes. When we talk about vision in neuroscience or in psychology, we're looking at this process that first starts from these things called electromagnetic energy sources. More often than not, in our environments, the primary electromagnetic energy source that we have is the sun. But there's lots of other electromagnetic energy sources that we've developed over the years. Anything that produces electromagnetic energy, heat waves essentially, is something that our eyes are sensitive to. Now, those electromagnetic energy sources can sometimes travel straight to the eyes. I suggest that you don't try this at home. If you try to look at the sun, you'll definitely experience too much electromagnetic energy to process it. But oftentimes, when we talk about vision, we're talking about the electromagnetic energy that's bouncing off of things. And when that electromagnetic energy bounces off of things, some of the energy is absorbed by those things that it's bouncing off of, and some of it's reflected. And what's reflected and can eventually reach our eyes is what we're actually seeing. And the properties to that electromagnetic energy that are bouncing off of those things has two major components to it. The first major component to this electromagnetic energy is the amplitude of that information. Essentially, it's how hard that energy is hitting our eyes, how compacted it is. If we're talking about amplitude and looking at it through a book, people often represent it as these kind of waves that are hitting our eyes in either big bounces or small little rivers or small little flows that are hitting us. This is actually a really bad representation of electromagnetic energy and the amplitude of it. I guess people can sort of get a visual approximation of it, but it gives us this impression that light is somehow snaking into our eyes. It's really not the case. Imagine essentially, if we're talking about differences in electromagnetic energy amplitude, being kind of the differences in beats. You could have a very soft tap that's hitting your eyes, essentially a low electromagnetic energy amplitude, or you can have really hard beats hitting us. That's essentially a high electromagnetic energy level or a great amplitude in this electromagnetic energy. And that high or great amplitude is really depicted by brightness. If the brighter something is, the greater the amplitude of the light hitting us. The frequency is hitting us at the same rate with small and great amplitudes, but the power behind it is what really determines how bright something is. The other component to vision is what we call the wavelength or the frequency of the electromagnetic energy pulses that are hitting us. Light travels, as you probably know, at a ridiculously fast rate. And when we're looking at the electromagnetic energy pulses bouncing off of things or coming from sources, not only that the kind of amplitude of these things is very strong oftentimes, but also the frequency of the pulses hitting our eyes is extremely fast. When we're talking about how we can differentiate between different frequencies in electromagnetic energy light, we talk about this term called nanometers, kind of the distance between the light particles or the electromagnetic pulses that are hitting our eyes. If we're looking at this differentiation in color or differentiation in wavelength of these things, The different frequencies or the different nanometers, distances between the pulses, dictates the different colors that our eyes are capable of seeing. Now, with the human eye, we're really limited in the distances between light that we can actually process. For most people, we have the ability to see light that ranges from 350 to 700 nanometers. Again, that's the distance between the different frequencies, or not different frequencies, the different light pulses that are hitting our eyes. But if you look down on this slide, you'll see that that visible light spectrum only covers a very small swath of the different electromagnetic pulses that exist in the world around us. There's other light frequencies that are hitting us at much higher frequencies and much lower frequencies. Now, mind you, when you're looking at this, if you see that small number of nanometers, say 350 or 500 nanometers, you might think that that's a lower frequency, but that's not actually the case. The distance is what's measured here in those numbers. So actually, your eyes are getting hit much more quickly with electromagnetic energy light when you look at, say, violet versus red, which is coming at us at a slightly slower rate because there's more distance between the pulses that are hitting us. So those really, really fast rays hitting our eyes that we can't see are gamma rays and X-rays. As it starts to slow down, it starts to become invisible again, and we get into things like infrared rays and radar and TV and other things where light is still passing through different mediums. It's just processed in a different way. Oddly enough, there are some species that do see a little bit lower on the light spectrum, a little bit higher on the light spectrum than us. But most vision is pretty much around what humans can see in that visible light spectrum. There's there's few exceptions even for certain humans, but just understanding that this is one major component to vision is a really critical thing that we need to know so we can start to understand how information, once it hits the back of our eyes, is then conveyed to the brain. And this is something that we do now want to turn our attention to. Now that we've covered what it is that we're actually seeing, how it is that we take what we're seeing and make a neural message out of it is the next big step in today's class. So once those electromagnetic energy cells hit our eyes, they travel to the back of our eyes, and we've got these specialized cells that can convert information on both the amplitude and the frequency of that light so our brain can start to process what it is we're actually seeing. And we don't want to start actually with the back of the eyes. There's actually a lot more to this process of vision than just what's going on in the back of our eyes for that neural message to be made. So in today's class, in addition to covering all the stuff we've already covered, we're going to also cover some of the major structures of the eye that you want to be aware of so you can understand this visual process much better. And when we look at the eye, what we see is there's actually a lot of critical players that are not only essential for vision, but to allow this eye to sustain itself and pick up on subtle nuances that we might sometimes overlook. If we're looking for a really critical structure to the eyes, most people tend to start with a structure that's not actually a physical thing at all. It's this thing called the pupil. The pupil is essentially a hole. It's the place in your eyes where light is passing through so information can get to the back of our eyes and we can start to process that information. Because of the fact that the pupil is just a hole that allows light to pass through, for all of us, our pupil is black. And that pupil will remain black our entire life unless something really bad is happening and light, for some odd, inexplicable reason, is actually traveling from the back of our eyes to outside, which is never going to happen for us. So if we're looking for the pupil, all you got to do is look into the center of your eyes, find that black spot, and you know you're looking at that gaping hole where light is passing through, so you're able to see all of the different things you can see. Around that pupil, though, is a really interesting muscular structure called the iris. And even though all of us have black pupils, we tend to have different colored iri. Some of us have blue ones, some of us have brown ones, some of us have green ones, some of us have kind of a weird gray combination. And there's lots of questions about why we have different colored iri. There's lots of postulations about how this might impact our vision, even though most research has suggested that if there is an impact, it's minimal at best. But regardless of the color of our iris, it's a critical player in how we're able to process information. Because electromagnetic energy is sometimes tough to process. It can be overstimulating sometimes when we get too much getting to the back of our eyes. So what the iris does is prevent too much information getting back to the eyes, but also allowing for the eye to take in more if there's a situation where there's kind of a lack of electromagnetic energy. You might have experienced this when you travel from somewhere that's very dark to somewhere that's very bright, or vice versa. When we're in bright environments, when there's a ton of electromagnetic energy that we could potentially process, our iris shrinks. It tightens itself up into a very small circle, which in turn makes sorry, I have to reverse there. Our iris, when it's bright, expands. It makes itself into a wide circle, and in doing so, in those bright environments, limits the amount of light that can pass through the pupil. In dark environments, as I was kind of alluding to earlier, the iris shrinks. It gets very tiny, and in doing so, it allows much more light to travel through the pupil and get to the back of our eyes. Now, the expansion process is something that actually happens very quickly. The shrinking process when we travel from a bright area to a dark area takes a lot more time. So it's sometimes tough to see that shrinking process. Expansion, though, is actually something you can experience yourself. In fact, I encourage you to, when you're kind of working through this, take a minute and go into a mirror that you have in your room or somewhere where there's a light switch and look into that mirror with the light off. Try to, with the light off, catch that pupil in your eye. And once you've seen it, once you've recognized the size of it, turn the light on. It works even better if the light is actually straight directed at your eyes. Not to the point where it's going to be harmful or painful, but try to have the light directed at your eyes when you turn it on, and you'll see something really cool. You'll see the immediate reaction of the iris. When that light goes on, the iris quickly expands or engorges, and it makes the pupil super small to help protect the eyes. It's something we don't often think of, something that we're not very attentive to oftentimes, but it is constantly active. Now, this is kind of neat on its own, but there's also some side things that psychologists have explored that have made it even more interesting over the years. One of the things that we've recognized since looking at the iris and its function is that there are certain things that can override this basic corresponding relationship between the iris and how much electromagnetic energy is present in the environment. One of the first things we recognized long ago was that the presence of specific chemicals in our system can sort of slow down how quickly that iris expands. Some of you might have experienced this in your life. If you've taken specific drugs, alcohol being one of them, there's a whole slew of other ones, though, that can have the same effects. It tends to slow down the reactiveness of the iris. It's why, if any of you have ever been pulled over at 2 or 3 in the morning by a police officer and they've shown that bright light in your eyes, you can kind of be sometimes caught not telling the truth, or maybe they can verify you are telling the truth when you tell them how much you've had to drink that particular night. If a police officer sees 2 giant black balls staring back at them as they're asking you how much you've drank and they're shining that light in your eyes, you have to understand they're not going to believe anything you say. But if your iris contracts very quickly, like it's supposed to when bright light is shown in your eyes, and that's a pretty good verification that that alcohol has not overridden numerous parts of your system, like that muscle in the iris. There's also some other things, though, that social psychologists have recognized over the years. One of the more interesting areas that we've gotten into recently is attraction and interest that we can sometimes gauge not through people's responses, but through people's physiological reactions to things. We now know that if you're interested in something, or you find somebody attractive, or you're just really engaged in something, you tend to kind of override the iris for a short period of time. And essentially, make the iris a little bit smaller, make the pupil a little bit larger so you can take in as much of the situation as possible. There are numerous researchers actually in various places that narrow in on your eyes when trying to understand what you're thinking and what you're doing so they can see how activated or how interested you are in something merely based on what's going on with your iris. It's a really cool thing, again, that we tend to overlook, but has a really critical role in our experiences. This is also true for another structure of the eye that's actually in front of the iris called the cornea. The cornea is actually a rigid structure that has about the consistency of your fingernails. And it's really there, not necessarily to do a lot with vision, but to protect all the structures that are critical in vision. You tend to, when looking at your cornea, have some bumps and bruises on it by the time you're a college student, because essentially when your eyes are scratched or when you're exposed to too much in the environment, like too much bright light, instead of getting damage to the other parts of your eyes, which are significantly more flimsy, that damage tends to be kind of directed at the cornea. And that particular damage that's incurred on the cornea is something that we can actually overcome. Now, you don't want necessarily constant damage to the cornea, that wouldn't be healthy, but we do actually want to be very grateful for having that cornea, because if it wasn't there, most of us would probably lose our vision by the time we get into adulthood, because behind the cornea is not only that muscle called the iris, but there's also a very flimsy structure called the lens, which we'll talk about next. And then behind that is this almost substance called vitreous humor, which has about the consistency of somewhere between jello and pudding. And if those areas get damaged, there's really no coming back from that. So making sure that those structures stay intact is really critical for our visual process. And the cornea helps us, well, at least to its best ability, ensure that those things are going to be okay, that those things are going to be intact. Unfortunately, though, when we do experience damage to the cornea, there can be some scarring, which could lead to the light getting to the back of our eyes being a bit distorted or having some imperfections in it. Now, some of you might be realizing right now, though, that you don't see those imperfections when you look out into the environment. And this might lead you to ask, well, how is that the case? What allows for that to happen, for us to have this fairly good image of everything in our environment if our cornea does have all these imperfections in it, even by the time we get into our teenage years? Well, to understand that process, I want to first turn our attention to another structure that's located behind the cornea called the lens. The lens is a critical component to our vision that we are always overlooking. The lens is this flexible, opaque structure that actually allows the light that we're seeing through our pupil to pass through it. But before it passes through it, the light is bent by the lens. And this bending of the light actually allows us to get the light that we're seeing at different distances to hit the back of our eyes at the same ratio or the same aspects. Essentially, it allows us to focus on things regardless of how far away it is from us. If something's very close, our lens bends, expands, and it allows that information to kind of get sent directly back to the eyes. If something's very far away, bends just a little bit more, and it kind of gets that light to be at least close to crystal clear by the time it hits the back of our eyes. For those of you right now that have vision issues, maybe you need glasses or you can't focus on things very well, know that it's probably not the fault of your lens. There are some situations where the lens doesn't bend properly, but for most of us, it's eye structure and other things that are really the critical players in the inability to focus on things clearly. And for most of us, the lens is constantly working and it's doing its best to give us the clearest image we can possibly get. We just are completely unaware of it. I mean, if you focus on something a few feet in front of you and then focus on something far away, both those things, as you're transitioning from one item to the other, will look crystal clear. The only way that's possible is for the lens to bend at an exceptionally fast rate, adjusting to all the different things in the environment so we can get that pseudo-clear image that's hitting the back of our eyes. There is, unfortunately, when we're talking about this process, a side effect to what the lens does. Numerous researchers over the years have shown through optics studies and just analyzing the biology of the eyes that the image that goes through the lens doesn't actually come out in the back of the eyes in the exact same form. Because of the structure of the eye, because of the structure of the lens, what happens is the image that goes through the eye is actually flipped and inverted. It's just plopped around. Of course, we don't see the world in this way. Right? Most of you don't think of the sky is up and the ground is down, and most of us aren't ever really confused as to which way is right and which way is left, unless, of course, you just don't have your right and left down very well. It's not, though, something that's a byproduct of us getting confused by what's hitting the back of our eyes. But if this image is flipped and reversed and these other things are happening, how is it again that we see the world as crystal clear? And when you combine that with what's happening from the cornea, most of us, when just looking at the basic elements of the eyes, would come to conclude that what's getting to our brain is a very convoluted, kind of messed up message that would take a lot to decode. How do we decode that? Well, this is something that actually puzzled researchers for a long period of time. So one of the first places they went to try to understand how this worked was developing a mechanism that kind of mimicked what the lens did. There were numerous people that invented these types of glasses that you could put on that would actually do the exact same thing that our lens does. They'd take the image that was hitting the glasses, flip them upside down, invert them, and have that image hit our eyes in that inverted way. And researchers wanted to see how this could impact people's vision. And there were a lot of fun games that were paired with it. Kids would play pin the tail on the donkey with these glasses on, or they'd try to play catch. And oftentimes in the beginning, when people would have these glasses on, they would perform, as you might expect, very poorly. But oddly enough, over time, if they wore these glasses for an extended period, they started to actually perform better on these tasks. In fact, they got better and better and better to the point where they were performing the same way they were without having the glasses on. And that might seem odd on its own, but what made it even more odd was that most of these people who had these glasses on reported that they started seeing the world as right side up, even though they were wearing these glasses. This was not necessarily an instantaneous effect. It often took hours or sometimes even days for this effect to occur, but for almost everybody, they got to a point with these glasses on where they didn't see the world the way it should have been seen. They didn't see the inverted world that the glasses were presenting to their eyes. Now, this was pretty cool. What made it even more interesting was what happened when these people took the glasses off, and suddenly the world was upside down and inverted. And again, it took them a while to recover and eventually see the world as right side up, but they adjusted. They were able to eventually see the world as it should theoretically be without any of the imperfections or the distortions that the lens and cornea were creating. So how did we do this? For a long time, it was just straight interjection as to how we were able to adjust to these things. Recent research in neuroscience, well, relatively recent research in neuroscience, has shown us that there's actually structures in the back of our brain that are involved in this course correction process. Right? Extremely active when something new pops up, when, say, an eyelash gets in your way, or when you do put these glasses on, or when you do have a cut to your cornea. And these things essentially are doing their best to try to figure out how to course correct and ensure that the image that our brains are processing is as clear as possible. And you can't blame this structure on your brain for any visual impairments that you have. It's usually doing the very best it can with what it has. But essentially, until everything is clear, everything makes sense, and this area is trying to find ways to decode the information in the best way possible. And this process is something that's constantly unfolding in vision. We've got areas that are constantly course correcting for different things that we're experiencing. And you can probably remember moments in your life where maybe you had, I don't know, an eyelash that was hanging down or a pimple on your nose or something weird that happened that made you see that thing for an extended period of time. But As time passed, you might have noticed that those things were no longer noticed. You didn't catch them in your visual field. It's not because they necessarily disappeared. It's usually because your brain overrid that information. It deemed that pimple or that eyelash is useless, not something that you needed to process. So it just kind of filled in the blanks. It figured out what it was supposed to be seeing and just kind of ignored that thing that was present or that new change that was causing activation in the eyes. This process is something that many neuroscientists are still kind of wrapping their arms around, trying to figure out how it works and how you can enhance it. But just for a class like this, it can allow us to appreciate how important not only structures in the eyes are for us to be able to see things, but how important the feedback is between the brain and the eyes when trying to make sense of this plethora of information that's hitting us at any given moment. Going back to the eyes, though, once that information has then passed through the lens, goes through the vitreous humor or the vitreous body, which is the structure that contains the vitreous humor, and it gets back to the back of our eyes finally, to this area that we call the retina. The retina is the area of the back of our eyes that contains these cells that do convert that electromagnetic energy into some type of a neural signal. But even when we get to the retina and we think we've reached the finish line, there are a lot of cool nuances to it that are worth mentioning. One of the things that's worth mentioning is that there is a subsection within the retina called the fovea. Sometimes it's mentioned as the fovea centralis. I like that name because it really gives you a sense of what the fovea is all about. It's the central portion to our retina. When light goes through our eyes, anything we're looking directly at tends to find its way to this fovea. The things that are in our periphery, the things we're not looking at clearly, can still hit our retina. It just isn't being directed to that fovea. And that differentiation between the fovea and the other parts of the retina is big when we talk about the next critical structures to our eyes, and these are these things called receptor cells. The cells that are actually converting that electromagnetic energy into that neural signal. If we look closely at the eyes under microscopes, what we see is there's actually a plethora of two different types of cells that are involved in this conversion process. Cells called rods and cells called cones. These are both very aptly named because looking at them under a microscope, rods look like rods, tiny rods sticking out of your eyes, and cones look like cones, traffic cones that have that circular structure on the bottom and kind of come to a point on the end. And not only are these cells differentiated in terms of their structure, but what they're used for is very, very different. Rods are located throughout our entire eye. They're actually very, very common cells that make up the vast majority of the cells in the back of our retina. But rods aren't necessarily very receptive to the different frequencies that our eyes are picking up on. So when we talk about differentiating between the different colors, our rods aren't really doing that. What they're doing is just picking up on the electromagnetic energy. And that's a really critical process in our vision that we tend to overlook. Being able to detect motion, being able to detect the presence of things, that's something that's primarily done by our rods. They're there to pick up on, well, what's there. But if we want to know the color of what's there, if we want to know fine details to what it is we're actually looking at, that's when we get to the other cells in the back of our eyes called the cones. Cones are very receptive to not only different colors, but daytime vision and fine details that we're looking at. And because cones are responsible for these things, they tend not to be dispersed throughout the entire eye. They tend to be centered in one particular area, the fovea, that central part to our eyes where when we're looking directly at something, we're kind of directing the light to that spot. And this kind of makes intuitive sense if you think about it. When you want to pick up on the fine details of something, and you want to detect the color of something, what do? You look directly at it. And when you're doing that, what that allows you to do is have your cones become much more active, much more involved in the visual process, even though they're not that prevalent in the back of our eyes, except for in that area. So we can catch all of those critical elements to what it is we're looking at. Now, if you ever want to see how this process works, there's lots of games you can play. One of my favorite is ones that have people walk kind of in people's periphery until they get to the front area with different colors. So you can hold different colors of paper, different balls, and see how very quickly you can identify the presence of somebody or something that they're holding when looking straight ahead and they're coming into your periphery. But it actually takes a considerable amount of time before you can detect fine details or colors in those things that you're seeing. Now, it's unfortunately a little difficult to play this game sometimes because we're often, even if we're not trying to, enticed to cheat and kind of peek over just a little bit to get some of that light to hit our fovea so we can process fine details and colors. But if you do play it earnestly, give yourself a chance, you'll really appreciate how your rods are picking up on stuff long before your cones are able to pick up on things. But once your cones get something, it can really help you understand exactly what it is that you're seeing. And we're going back to this process of vision, though. Know that rods and cones are, again, doing the same general thing. They're collecting that electromagnetic energy so that information can be sent to your brain. For it to get to your brain, well, there has to be an out spot. There has to be a spot where the information is leaving your eyes, heading down some neural path to the back of your brain in an area called the visual cortex, something we talked about earlier. What is the out process? Well, the out process is this area called the optic nerve. It's a small little space in the back of your eyes that allows the light to actually leave. There is a consequence to this optic nerve, one that we often don't recognize and often can actually be accounted for in most visual experiences. That out spot has this kind of weird thing that comes with it called a blind spot, where we actually have no rods or cones in that structure or that portion of our retina where the optic nerve is located. Again, most of us never notice this little blind spot because we have two eyes and the other one tends to account for what the other eye is missing. We've also got amazing memories that allow us to fill in the blanks even if we're just looking at things with one eye. But if you want to detect the blind spot, there are ways to do that. If you look at your textbook, you'll see in the textbook an image of a lion tamer and a lion. And what you're asked to do to be able to locate your text box, or sorry, to locate your blind spot, is put one of your eyes above the lion tamer. In this image that you see in front of you, it would be putting your right eye in front of the lion tamer and then slowly back away with your eye consistently centered over the lion tamer. And what should happen for you as you're doing this is you'll see the lion tamer with the lion tamer with the lion tamer with the lion, and then eventually you'll get to a point where the lion just disappears. Essentially, that lion is disappearing because you've aligned that lion with your blind spot. And you do the same thing with the left. You just have to flip the book around and have the lion on your left side. And again, you'll back away and eventually the lion will disappear and it'll look like a whole circle instead of a circle with a lion in it. And that's a byproduct not only of our blind spot, but of our brain trying to fill in the blanks. You see a consistent circle and it just kind of fills in the blanks even though it knows there's a lion there, and it gives you the image of a full circle with a lion tamer when you hit that blind spot. Really neat to do this because it helps you appreciate that you do have a blind spot, but also it helps you appreciate that your brain is constantly, even when it kind of knows what it should be doing, trying to fill in the blanks to make things look as clear as possible, despite, again, these imperfections in our vision. But if we're looking at the visual process, now, this blind spot is critical because, as I mentioned, that's the path out of the eyes. Once our eyes pick up on that information, it starts heading to the brain. But before it gets to the back of our brains, to that visual cortex or occipital lobe that we've talked about earlier, there's a couple stops that need to be taken along the way. The first stop is a really important structure called the optic chiasm. This is the structure in our brain or in our nervous system where the information from our left and right eye first meets. It's also a structure in our brain that's really critical in this filtering process that we talked about a little bit earlier. We discussed when looking at the parietal lobe and partially the temporal lobe, this idea of something called contralateral communication. One side of your brain tends to be linked with one side of your body. Oddly enough, it's the right side of your brain linked to the left side of your body and vice versa. Well, this contralateral communication also works for vision, but it works in kind of an odd way. It's not that only your left eye information goes to the right side of your brain and only your right eye information goes to the left side of your brain. When we're talking about vision, we instead focus our attention on these things called visual fields. This is always a very difficult topic for people to process. But if you could, just look straight forward and imagine, as you're looking straight forward, an imaginary line kind of extending from your nose out. And everything to the left of you, as you continue to look straight forward, you can't look to the left or you screw it up. As you continue to look straight forward, everything to the left of you is part of what's called your left visual field. As you continue to look straight forward, everything to the right of you is a part of what we would call your right visual field. The information that's in your left visual field is actually being processed by both your left and right eye. A little bit more by your left eye than your right eye, but both your left and your right eye are processing information in your left visual field because that light is coming from the left side and it's hitting, oddly enough, the right side of both your left and your right eye. Imagine light kind of coming as a straight line, working its way through the pupil, working its way through the cornea, working its way through the lens, all those structures that we talked about, and then straight lining it back to, again, if we're talking about our left visual field, the right side of both our left and right eye. Well, that information that's hitting the right side of both our left and right eye is, again, left visual field information. And when it reaches the optic chiasm, oddly enough, that information hitting the right side of our eyes goes to the right side of our brain. So we talked about our left hand being controlled by the right side of our brain. Well, our left visual field also goes to the right side of our brain. Our right visual field has the exact opposite effect, where everything hitting the left side of our eyes, coming from our right visual field, eventually goes to the left side of our brain. That's depicted in this picture, if you're going back to it, by the pink lines and the black lines being information that's coming from our right visual field, and the pink lines being the information that's coming from our left visual field. That contralateral communication flip happens in the optic chiasm, and it's really critical for contralateral communication with the body to work the way it's supposed to. And if we want to control our hand and catch something that's coming on that side, and we want our brain and the two areas responsible for movement and vision to be very integrated with each other. So if we're moving our right hand to catch something coming from the right side, it's essentially a volley between our occipital lobe and our frontal lobe / temporal lobe that allows us to coordinate that movement instead of having to cross through all of the different parts of the brain to coordinate all of it. It's just a volley of information happening, if we're talking about the right side, of information happening on the left side of our brain that allows us to do that. It's a really beautiful system that is somewhat complex in design and tends to overwhelm students. But if you slow down and you look at it, you'll be able to recognize, once you kind of get how it works, that this is the peak form of efficiency that we can devise. That information that is split in that optic chiasm is really there for that process. But before the information that's been split goes to the brain, there's actually one last stop along the way that's worth noting. It's an area of the brain that's critical for pretty much all of our senses. It's a structure called the thalamus. Consider the thalamus kind of like our gatekeeper for sensation. There's a lot of different processes that are going on at any given moment and any given time in our day. We're constantly being bombarded by touch signals. We're constantly being bombarded by sounds and visual stimuli. And if our brain were to try to process everything, we would be completely overwhelmed. So how do we dictate what it is we're going to pay attention to and what it is we're going to kind of ignore? Well, one of the big players in this process is our thalamus. The thalamus essentially filters out what's critical and what's not. There's a lot of interplay between the thalamus and, say, our frontal lobe, which is helping us decide what to pay attention to and what to ignore, and also the different parts of the brain that are critical in processing different information. It's a very involved process if you really want to break it down to the nanoseconds and all the different things that are going on in the brain. But if we're looking at where the information tends to get blocked, most researchers have consistently argued that the thalamus is that filtering spot. So not all information will go from the thalamus to the back of the brain. A lot will be kind of cut off at that point. And it's only the critical information through a whole cascade of communications that we've determined is critical that reaches the back of our brains to that area called the visual cortex. And that, as we mentioned, is where the real heavy lifting starts to take place, where we start to pick up on things like color and angles and exactly what it is we're seeing in different spots of our eyes. In fact, if we got into neuroscience and looked at our visual cortex a little bit more closely, you'd see that the back of our brains actually have all these different grids, these different structures that can help us pick up on information coming from different eyes, colors of that information, how persistent it's been, and that's just the first steps in the process. It's part of an area in our brains called V1. You move forward to another structure called V2, V3, other types of processing take place. In fact, you could dedicate a couple weeks or months in a sensation and perception class just talking about that perceptual process that starts to take place in the back of the brain. As I just mentioned there, that's when we do start to get into the next step in the process, perception, and decoding that information that's now reached the brain. And that's what we're going to actually be covering in our next class. So for today, this is kind of the end, the finish line for what we're going to be covering in that sensation process. In our next class, I want to make sure that you're well prepared, so be sure to do the readings. And when you get through those readings, you're going to get to see a lot of really cool things and examples of how perception works, but also you're going to see a lot of ways that we've kind of discovered this process through different techniques and tricks that we can sometimes play on the brain. So for now, I hope you enjoyed this. Make sure you do some studying, do some reading, and I'm sure I'll see most of you very soon when we get into that process of perception. Take care.